Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Quick note before we begin, the Finding Genius Foundation, as part of the Finding Genius Podcast, has recently completed a book about understanding viruses. So the creation of this book was to interview 100 virologists, ask them a lot of deep, difficult questions, take the most difficult questions, and then re-interview the top 25 or so and ask them the hardest questions I could think of. And we compiled that all into a book. So you'll see question and 4 or 5 experts' answers. Question, 4 or 5 experts' answers. There's about 30 questions in the book. I think it's a great read for the layperson and for the researcher. It talks about a lot of speculation in the world of viruses, such as are they alive or not, and why is it important? Why do viruses go latent or hidden or ineffective or sit in a person or an animal or another creature for weeks, months, years, and then suddenly become virulent and affect that person? So there's a lot of really provocative questions in the book. It's now on Amazon. So if you go to Amazon and type in Finding Genius, you'll see the book on viruses. It's also on Kindle. The Audible version is in production and should be ready in approximately a month. But if you want to go and order it now, you can do so again by going to Amazon or Kindle, or go to findinggeniusfoundation.org and go to publications. There's an opportunity as well to get the transcripts of all the interviews and to hear the original interviews themselves. If we put them all together, the book would be about 1,000 pages, but we condensed them down to make it juicy and concise and tight and very interesting. So I hope you'll check out the book. We're now working on one about cancer, but this is going to be our goal is 3 times a year to come out with these masterclass books that I think will inspire new scientific research, and I hope you'll check it out. Thank you.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now a part of the Finding Genius Foundation. I have a returning guest, very interesting, very cool guy, Doru Paul. He's an associate professor of clinical medicine at Weill Cornell Medical College. We're going to talk about the cancer book that I'm putting together. As listeners, you may know that I put together one on viruses. It's now on Amazon and Kindle and soon to be Audible. If you go to Amazon or Kindle and you type in Finding Genius, you'll see the book. And I try to ask very provocative, difficult to answer questions about viruses and still make it accessible for the public. So you can check it out there. Today, again, we're working on now the cancer book. So Doru, thank you for coming.
Thank you for inviting me.
Yeah, so let's jump in. Do you see that cancer is a self-sufficient, independent living organism inside of a person, or do you believe it's just essentially a corruption or a bunch of cells acting individually, or do you think they're acting again in concert and they constitute a separate living organism?
So I've been pondering a lot about this question, and the model that I'm considering now, it's a model in which there is a continuous interaction and communication not only between the cancer cells themselves, but between the cancer cells and the tissue where they appear, and also between the cancer cells and the whole organism. So I do not see cancer as solely a disease of a cell that is running crazy, or even a disease of a tissue, but I see cancer really like a disease that appears in certain organisms that will prepare the soil for cancer, the macro environment, as opposed to the micro environment for cancer. And the reason I'm saying this is that there is data from the longitudinal studies that looked at transformations in the blood of individuals over time. There are studies like that going over 20 years in which different metabolites were drawn and analyzed from the cancer of people. And then when they develop a certain form of cancer, you can go back and identify changes in their blood that happen 2-3 years before the cancer development. So what I'm trying to say is that in order for cancer to appear, you need a certain macro environment. The organism itself is giving, in a sense, birth to cancer. It's not disease solely of the cell, and it's not only the cell which is deranged in some way that this genetic theory is saying, but it's really a combination of factors at the cellular level, genetic level, and epigenetic level, at the tissue level with changes in the tissue, and at the macro environment, that's the whole organism.
So, I see it as a forced continual maladaptation due to, again, continuous metabolic or otherwise stress in a given tissue. And the cells are trying to adapt. They're undergoing epigenetic change. Their microbiome, their localized microbiome is probably changing. But at some point, the adaptation, I would guess, runs into a brick wall, and they de-differentiate in the hopes of, again, surviving. And this is what leads to cancer. That's my thought. What do you think?
You can certainly think in these terms. I've been calling this maladaptations different programs. So what I think is cancer is not solely of a disease of a disorder or a disorder disease. So when you're seeing all these aneuploidies, all these chromosomal changes in numbers and quality, or the genetic changes, the mutations, behind this, there is still a living entity, the cancer cells. They are very different from the normal cells. This is true. They have a different number of chromosomes, different qualities of chromosomes, but they are still alive. So behind this, there are some programs that are keeping them alive and functional. And as you say, this is a form of misadaptation to some type of situation that may be, stress at the level of internally at the level of the genes, or externally at the level of the tissue. But you can certainly think in terms of these programs which are responding in a maladaptive way to these changes.
What do you think happens first? How does cancer literally start from the beginning? Do you think there's only one way, or there are multiple ways? And again, what's the order of new attributes that are acquired by cancer cells?
Yes. So your question is really, how does cancer appear? What is the first cell or the first cells that are making cancer? First of all, let's start with what is known. We know that there are some mutations that are present in the genes. Are these mutations the cause of cancer? It's a very strong no. Why am I saying that? Because the same mutations, let's take, for example, the case of melanoma, they're also present in normal nevi. The BRAF mutation that's present in melanoma cells, it's also present in normal nevi cells. The TP53 mutation that has been also present in more than 50% of solid tumors, it's also present in some cases of rheumatoid arthritis. So the genes per se are not the explanation of cancer. Is it one single cell that creates cancer, or there are several cells? Nowadays, becoming fashionable this theory of polyploidy, that there are several cells that are giving birth to cancer, similar to this morula that is developing in the embryonal life, like this grape structure of several cells that appear due to some stress conditions. This is really difficult to demonstrate that the origin of cancer is this morula. There are studies that are showing that if you take some cells and then you expose them to continuous stressors like chemotherapy, at a certain point in the number of divisions, you'll see the polypoid cells appearing. But these are cells that are exposed in vitro to this type of stressors. On the other hand, in vitro, finding these polypoid cells as the origin is not as simple, and to me, it's not really clear that the origin of cancer stays in these polypoid cells. Other ideas have been there, you have an embryo that develops, so it's a fusion between two cells that are going to develop cancer. But again, if you look at the number of chromosomes in cancer cells, not all cancer cells are aneuploidic. They have extra numbers of chromosomes. So the fusion idea, it's also clearly not universal. As you said, it's also possible that it's not only one way to develop cancer, that there may be different mechanisms behind that. So it's not only the polyploid theory or the stem cell theory, which is another theory that has been popular, but it's not really been demonstrated consistently in solid tumors because there are other cells that can be at the origin, which are more differentiated than the stem cells.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
So clearly, in different types of cancers, you may have different origins. What is striking, though, despite these differences, is that the phenotype of cancer, the division which is uncontrolled, and the invasion and the metastasis is really common to many cancers, many different types of cancers. Now, is this commonality like a common evolutionary effect? It's difficult to think that evolution has really randomly selected for this type of cancer phenotype. So I really don't think that cancer per se is selected by the evolution, because I don't really see the advantage for a particular organism to have a cancer phenotype selected, during evolution. So this being said, immediately, because it's not selected during evolution, you can also think that the origin of cancer is not really in the mutated genes or the aneuploidy or things like that, but it may be, in fact, a combination of factors. Majority of cancers develop after age of 50. 90% of cancer, they develop in elderly individuals, and maybe the systems of control that are present both inside the cells, in the tissue, and also at the organism level, like, for example, the immune system, after age of 50, they are really starting to fail, and the apparition of cancer may be explained like this type of perfect storm that happens at different levels, at the cellular level, at the tissue level, and at the organismal level. This being said, I will also go into what is the primum movens. In order for the cells to divide, you need energy. And Otto Warburg has been, introducing this idea that at the origin of cancer, you have mitochondria dysfunction behind that. There is some type of abnormality in the mitochondria, and this is why the oxidative phosphorylation does not function, and you have the Warburg effect, which basically produces ATP in low numbers in an ineffective way to produce energy. So,
quick question here: If we look at the hallmarks of cancer paper from 2000
yes,
and then the recap in 2010, there are hallmarks that are common to many cancers. So what do you think the order of the appearance of the hallmarks is? Is there immune defense first? Is epigenetic change first? Is mutation first? What's next? Any insight into why and what comes first?
Yes. So really, I think that the primum movens, it's the organism. So I consider cancer a disease that's a systemic disease to start with. So a decline in the immunity that is happening, with age, with stress, with different other factors, I think it's at the origin of cancer. So you have transformations with age at the tissue level. You have these mutations that, I just described that are present. And then you have this type of perfect storm, as I said, between these influences from the decline in the immune system. You have the presence of some mutation. You have then the tissue, which is affected, is not controlling the local growth of cells. So the communication between the cancer cells and the normal tissue is defective. So this is what in my mind is the explanation. So I don't really think that the origin of cancer, it's a sole hallmark, like, for example, the division. Benign tumors, they have division that is uncontrolled. Cancer cells, they are not benign cells. They have other hallmarks besides just cellular division. If you take the essential hallmark of cancer, which is, in my eyes, is metastasis, there is not something similar with oncogenes and repressor genes responsible for metastasis. The metastatic process, it's mostly an epigenetic process. And in terms of genes, there are some genes that are responsible for the metastatic phenotype, but these genes are not mutated.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
These genes, they have some inhibition of their function at the epigenetic level, but there is no mutation there. So in terms of what Weinberg and Hanahan were saying in the initial paper or the subsequent paper, that you have, the genetic instability, or you have the immortality or certain changes in the metabolism, I think these are consequences and these are not the root of the disease itself. So the root of the disease is not in my eyes going to be found in only genetic changes or only epigenetic changes. I mean, just think, in order for a cell to divide, needs energy. This energy is coming from really the mitochondria and the metabolism. So you can think of cancer as being also a metabolic disease. Now, is it solely a metabolic disease? I don't think it's solely a metabolic disease. I think it's a combination of metabolism, epigenetic changes, and genetic changes. What is fundamental in all these three? I believe that fundamental is really the metabolism. Why is the metabolism? For a cell to do different things, to divide, to invade, to go a distance and then grow again, you need energy. So to me, cancer is a disease of energy and a disease of communication. These are the two, I would say, key factors for cancer.
Why do you think cancer cells have tropisms for certain organs to metastasize to?
There have been several explanations for the tropism. The classical explanation was the mechanical one, that, you have certain capillaries with fenestration, so that's why cancer cells, they can go in the liver, they can go in the lung. If you take the 10 most common tumors, you will see that the metastases, they mainly go to the lung and the liver. So one idea is that in the liver, you have more nutrients, and they go there because, they want to grow, they want to basically form colonies. When I'm saying once, of course, I'm humanizing cancer cells, so they're attracted by nutrients in these locations. Another very intriguing and interesting theory is the theory of Dr. David Leiden that identified on the surface of exosomes, this nanometric particles secreted by the cells, a code, like a postal code that tells where to go, like a sort of protein GPS system. So he found that a certain code will direct cancer cells towards certain metastatic locations. Like, for example, you have this integrin code for bone mets, another integrin code for liver mets, another code for brain mets, etc. So he has developed this theory of precise metastatic development due to this exosome code. And this is how he explains the Paget hypothesis that certain cancers go to certain organs, like prostate, for example, going very often to the bones, or lung cancer going often to the brain or to the liver. So he has been describing transformations of the surface of these exosomes that are related to certain places where cancer grow. Another idea that has been also described previously by the Masage group, Joao Masage at Sloan, was that there are different genes that are activated for certain location of cancer. But again, the metastatic process is not mainly a genetic process, so this theory of certain genes related to certain metastasis destinations is not really, I would say, proven. So this is what it is. Now, in order for the cancer cells to grow in a certain place, besides the cancer cells going to that place and being directed to that place, you need also other stromal elements. And an interesting idea is this triangulation between the primary tumor, the bone marrow, and then the distal site. So with other words, in order for the metastasis to happen, besides directing the cancer cells to specific sites with the help of these exosomal patterns, you need also the help of these bone marrow-derived cells that are going to help really the distal development. Besides this being said, and the origin of cancer cells and things like that, there are other theories, very interesting theories. One theory is that when the cancer cells metastasize, it is not solely one cell or several bunch of cells like in a grape structure going through the blood and metastasizing at distance. One interesting finding is that sometimes cancer cells, they go at distance with a bunch of transformed fibroblasts that will help them to go there. So the things are not as simple that we're saying, okay, this exosome code and that, or you have, the bone marrow. There are different probably systems, and in different cancers, there are different mechanisms for that.
Well, speaking of exosomes, what kind of communication do you think is going on between primary tumors and metastases? Is it like a master-slave arrangement? Is it a coordinated action amongst all different tumor sites? What are your thoughts?
It is clearly, the communication exists for sure. If we go back in 1996, 1997, with experiments with this angiostatin drug that has been developed. At that time, the experiment looked like that. You take 10 mice, and in these 10 mice, you establish some lung tumors, and you take other 10 mice, you establish also for control lung tumors. And then when you're taking out the tumors, you will see metastases appearing in a distance when removing the primary tumor was associated with new metastatic growth. And then in the mice that you're giving this angiostatin that was identified initially from the urine of mice, the distal growth does not happen. It was really like in all 10 mice in which angiostatin was given, which is basically vascular endothelial growth factor inhibitor. The cells were not growing at distance. So what was clear is that the primary tumor was secreting some type of really molecule, and this is what angiostatin was, that is inhibiting the growth of metastases. So this was a formal demonstration of communication between the primary tumor and metastases. Now, it's also the opposite. This was, the famous experiment of Judah Fallman that did it in the 90s, and then this molecule really never became standard of care in the United States. We had, instead of this, we had the VEGF direct inhibitor, Avastin, that was developed here. And the molecule, interestingly, the angiostatin molecule was interestingly purchased by the Chinese, and it is standard of care in China in lung cancer. But this is an economic discussion. So communication, another idea. When you take out the primary tumor, in some cases has been well described in kidney cancer, there was also regression of metastases at distance. So here you say the exact opposite phenomenon. So in the example of Judah Fallman, you had taking out the primary tumor, you had the development of metastases because the primary tumor was secreting this angiostatin that was inhibiting the growth at distance. In the case of kidney cancer, taking out the primary tumor was in fact making the metastases disappear. So you can see that this communication is not one way. There are different ways of influencing. Also, there was another interesting concept that the metastatic cells, they can reseed the primary tumor. So once they're at distance, they can come back to the primary tumor and also help the development of the primary tumor. So this ongoing interaction and communication between the primary tumor and the metastases, it's very interesting. And I've been thinking as a sort of network that forms between the primary tumor and the metastases, like a malignant network in which they influence each other. Again, they may influence each other in both ways. The primary tumor may stimulate the development of metastases, or they may inhibit it. And the opposite, the metastases, they may stimulate or inhibit the development of the primary tumors. And then, of course, you have development of second metastases, both for primary tumors and from the first metastases. So it's really a complex network of interactions.
So you think there's definitely, do you think there's an emergence, again, as like if I look at cancer and there's just a few cells that are cancerous, how much coordination is there going to be versus 1,000 or 1,000,000 or 1,000,000,000 in a tumor versus main tumor versus, and in addition to metastases? Like, what do you think the emergent properties of this, I'm going to call it an organism, are at various stages of its development?
When it grows initially, it is mostly the interaction with the local stroma. At a certain point, when you have this metastasis developing, then the system becomes more complex. It's also like in the development. So you have initially, I mentioned this morula. You have the egg that is growing, and in the initial morula, and then you have the blastula. So you have different stages of development. You can think of cancer, some type of malignant embryo that's developing. There is, pregnancy. It will be like a metaphor for cancer, to understand cancer. So you can think that like the whole body is a sort of placenta that is making this cancer grow. So you have the interaction between the newly formed organism and the whole body, which is the placenta. So you see that the interactions are in both sense. Initially, is the placenta that that feeds cancer, but at a certain point, this malignant embryo starts influencing a lot the body in which it grows, and it's making it really very, very sick. And ultimately, this is why the patients die, because of the influence that the cancer will have on the body that made it grow initially. So these interactions, I think they're there. I mean, I would not call it unicellular form of life or a multicellular form of life. I would call it more like a dyskaryote. Like you have eukaryotes, prokaryotes, dyskaryotes. So you have a different form of life. Why is it different? Because if you look at the number of chromosomes, it's not maintained, right? So you cannot say that it's something like plant with the same number of chromosomes, or like an animal with the same number of chromosomes, or a being, because the number of chromosomes is variable. So it is really a very different behavior, and this form of life is not a xenobiont, is not really like coming from the planet Cancer in the body and developing this weird stuff. No, it's the body itself that it's creating this type of abnormal dyskaryote life form. So I would say that it is a different life form that may be even different from the prokaryotes and the eukaryotes. So of course you have multicellularity because you have multiple cancer cells, but the behavior, it's both belonging to the eukaryotes and the prokaryotes. So I wouldn't call it like some type of atavism in terms of monocell, like a single cell, or an atavism of a multicellular organism. But I would think of it as a different life form.
Yeah, that's what I was wondering. Okay, but at different stages of its development.
Yes, absolutely correct.
Are there any particular emergent properties or characteristics that stand out to you that amaze you or surprise you? Does it tend to evolve any abilities that surprise you?
Yes, absolutely. So one thing is, how fast does it develop, right? And you have this idea that it grows over years and it's becoming centimetric when you have some type of like a vascular development. So you need vessels to grow beyond the 1 cubic centimeter. But in my experience, because I'm a clinician, I've seen growth and the metastases happening within days. So it is not as simple. It is not, the behavior is not homogeneous. I've seen a particular patient that was receiving radiotherapy, and during radiotherapy, literally the cancer started spreading, and we are talking about really days and less than a month. So the evolution and the behavior of cancer is not something that even an accelerated evolution cannot really explain things that are happening so quickly. So we definitely don't know everything about cancer's behavior, and I will call it really metaphors. The fact that we're thinking of cancer as some type of accelerated evolution or some type of development problem.
Or regressive evolution, yeah.
Right, correct, yes.
Do you know of anyone that's looked at neoplasms and compared that to cancerous tumors and to look for the differences? Because I would think you'd find some interesting commonalities or differences.
Yeah, absolutely. So one thing that we discussed, so a neoplasm is really a new growth. So you can have benign tumors. So what you're basically alluding to is the fact that you have a benign tumor and you have a malignant tumor. So what is the difference between the two? Well, the benign tumors that don't really break membranes, that don't invade locally, they don't spread at distance. So if you're looking at different aspects in terms of genes, again, you can have benign nevi that have the same genes. I mentioned the BRAF genes, which are similar, or the NRAS genes, which are similar to the melanoma genes. So at the genetic level, they may be similar. What is really, I would say, more specific for cancer is also this Warburg phenomenon, because in general, the anaerobic glycolysis is really linked to cancer. If you're going to compare, overall the benign tumors versus the malignant tumors, in general, the malignant tumors, they tend to have this Warburg phenomenon. So metabolically speaking, they are different. Epigenetically speaking, they are different. And also, I alluded to these genes that are repressed, the so-called metastatic genes. Again, this repression is probably related in particular to the malignant tumors and not to benign neoplasms.
Yeah, I just didn't know what if it's been studied, what had been learned. But how about the heterogeneity of tumors themselves? Has anyone tried to 3D map the heterogeneity and characterize it within a given tumor, and then trace back the clonal lineages to see, can you reconstruct using a computer how it formed, what came first, and how it moved around?
Yes. So these reconstructions have been done. Charles Swanton looked at this so-called trunk mutations versus branch mutations and looked at what was common between different clones and looked at the mutations that were common between different clones and went back to see which are, the first mutations that are there. So this has been certainly studied and done. How can you use this information? One idea that Charles Swanton introduced is that maybe you can find neoantigens that are common between the different clones, and then you can have this targeted by either some type of intelligent T cells or some antibodies, adoptive T cell therapy or CAR T cells or other ways to target these neoantigens that are present on the trunk mutations. So related to the trunk mutations. Now, the heterogeneity is important. And this is a very interesting point, because initially we learned at school 30 years ago that cancer cells are like a school of fish that are all the same. Nothing that can be, I would say, more far from the truth. Now we learned with all this single cell analysis that every single cell in a tumor has different mutations, have different epigenetics, and cancer is a totally heterogeneous environment. The tumor environment is very heterogeneous. And for example, in certain cancers, there have been analyses in terms of heterogeneity that looked really to see if a more heterogeneous tumor in terms of both stroma and immune cells and cancer cells as a worst. And there have been a study that looks specifically at that, and it appears that at the tissue level, when heterogeneity is present, the prognostic tends to be worse. And this has been computed using the Shannon index, the same as Shannon with the information theory. So you can really measure the heterogeneity in the tissue really in a formal way, as you say, via computer. You can really compute this type of heterogeneity there.
Okay, so it's been looked at, but again, have they been able to reconstruct from the beginning how a tumor grows and, again, what clonal lineages arise and things like that?
In vitro, research has been done mostly with some type of petri dishes where you put some cells, they grow, and then Of course, you have all these xenografts you're putting under the skin of mice or other lab animals, some cancer cells, and they grow. And recently, you have this different ways of growing cells on some tissue mimicking environments. So you're having the organoids that have been developed, initially by research in Netherlands with colorectal cancer, and then now we have it in different cancers. So basically, you have really the growth of cells trying to mimic as much as possible the growth in a tissue. So we're going from like petri dishes to these organoids, but still, this environment, it's a totally artificial environment compared to the natural environment, because in the organoids, you don't really have immune cells. They're trying to develop now organoids with immune cells inside. And it's really just like a scaffold on which the cancer cells grow. And you don't have all this variety of environmental cells. Like, you have the fibroblasts, you have macrophages, different immune cells, like more than 30 different immune cells. So the in vitro research is really very far and still in infancy compared with what happens inside the organism. So in terms of 3D, if you're looking at what's going on, you will never find two cancers that are the same. And not only that, but if you're looking between a cancer and the metastases, also the patterns of growth are going to be different. So by different, I mean, if you're looking to compute how many cancer cells are there, how many stromal cells are there, how many immune cells are there, you will find a large variety. You'll have, for example, in pancreatic cancer, some pancreatic cancer, they have like 10% of cancers, and then everything gets inflammation with inflammation cells, with modifications in the stromal cells, and the cancer cells, there are very few. And you can think also of the fact that some of the symptoms of cancer are not really due to the cancer cells per se, but to the abnormal response of the organism. Now in the COVID era, we've been seeing all these people, in the ICU dying because of all this inflammation that is developing, and then we've been trying to fight it with steroids and anti-TNF agents. So something similar happens in cancer. Sometimes, there's a lot of inflammation there, and some tumors are very inflamed. Also some breast cancers, this presentation with breast cancer that look really like a wound. So they're very different. And if you look in the 3D, the differences are really striking. So you cannot really find two cancers that are the same.
Yeah, but there would have to be some similarities, or it's not completely heterogeneous or completely a mess. Like,
yes, so
even if you look at the structure of a tumor, I mean, they're probably like Pablo Picasso, but they're not completely random, right?
Well, they're not completely random. And if you would want to know an analogy, you'll have on one hand something that it's like, some type of like smoke, which is totally random. And then like a crystal that is completely organized. Cancer is somewhere in between. Is it like fractal in a way? Does it have regions of order? It is not really fractal. It's like, there was this chaos theory that was introduced, I would say, something like 20 years ago. So it's like on the edge of chaos. So it's not really chaotic. It is not organized, but it's on the edge of being disorganized. So it's in between. And this is what is striking. So there is some organization there, but it's not this type of like, complete organized system, like, as I said, like a crystal, something very organized, like a lattice, like a organized network. It is not completely disorganized, like totally random. You brought the Jackson Pollock, which is a very interesting comparison, because the Jackson Pollock paintings are not completely chaotic. They have been looked also at, using, I think, also Shannon Index to see what's the organization of a painting by Pollock and why does it appeal to our eye. And it is not something which is completely chaotic like a jungle, and is not something completely flat like a tundra, but it's something more like a savanna, and you have some trees, so the Jackson Pollock paintings are, I think, a very good example for cancer, which has... Well, also like Pablo Picasso, yeah.
He had pictures of people and things, but they were blocky and distorted, but you can still tell, that's a woman, that's a table, it's a dog,
Absolutely correct. So yeah, I mentioned, yes, I mentioned Jackson Pollock, but Picasso is another example. Absolutely correct.
So what, if you look at primary tumor versus metastases and you characterize their heterogeneity and their structure and their, Jackson Pollockness, what's noticed between primaries and metastases? Has anyone done a study where they've looked at groupings of hundreds of them in many different people to see if there's a similarity, at least amongst the primary and metastases?
The pathologists are looking, when we have doubts, they're comparing the primary tumor metastases. And in general, probably I would say maybe, giving a number, maybe 80% of the metastases, they tend to have some similarity to the primary tumors. But this is not always the case. And what is really interesting is that this theory that cancer develop in a really linear way, you have some dysplasia initially, and then you have some more mutations leading to some local growth, and then you have some invasion, like local invasion, and then metastasis, is probably completely wrong. So what happens if you take a tumor, probably you're going to have some cells that are more, cells that divide, like they have this program of division activated, some cells that have, the program of invasion activated, and then some cells that have the metastatic program activated all at the same time. So it is really, I would say, a paradigm shift to think of cancer not as something happening in a stepwise fashion, but something which happens in parallel. That these things, the development of the primary tumor and the metastases, they are happening simultaneously. And these are factors that are probably tissular factors and also organismic factors that influence the apparition and development of metastases. Also, if you look on the scan, talking about the metabolism, you'll see differences. So a primary tumor may be, having some hypermetabolism, but the metastasis may have less. Or the primary tumor may not have so much hypermetabolism, and then the metastasis may have more. So also what they consume, if you're looking at the nutrition of the cancer cells, they may be also different in the primary tumors and the metastases. The primary tumors may eat more glucose, metastases may eat more glutamine or more amino acids for the primary tumors, and then more glucose for the metastases. So you have different combinations of this, and there is a clear heterogeneity, both at the genetic, epigenetic, metabolic level.
What if you took electronic health records of millions of people and you could see which ones had liver cancer, pancreatic cancer, et cetera, and you looked for commonalities, do you think anything would be observed?
Yes. So if you take liver cancer is very common in Africa. And of course, it's common there because you have more hepatitis. So you'll find this commonality. So what I'm trying to say is that there are common things. There is this type of external environment that is transforming into an internal environment that subsequently is leading to cancer. So, alcohol in combination with smoking produces a risk of 30 times more of esophageal cancer. Then you have the BRCA genes that give a risk to women that have these mutations in their genome of 50-60% of breast cancer. You will find commonalities. Yes, if you're going to take and look at what was going on with those patients, you'll find things in common. You'll find things that are common in terms of the diet, things in common in terms of the environment in which they lived, things in common in terms of the history. I just mentioned the breast cancer. So it is definitely not random. So that certain cancers appear in certain individuals. This theory that was discussed, at length with this type of bad luck, I completely disagree with the bad luck theory.
Okay, yeah, I just figured it was another way of looking at things. If anyone's done because you could have access theoretically to millions and millions of people's, electronic health records. So I think it would just be a very valuable source of data.
Totally right. I mentioned that longitudinal studies that were done and finding that you can identify changes in the body long term that the actual cancer develops. Also, you can look retrospectively at different factors. I did such a study in which I looked at what was the long-term survival with this new epidermal growth factor receptor inhibitors, Tarceva, to see how they change the landscape of lung cancer and how they change the 5-year survival. What other factors are important for that? And to my surprise, it was not as much these new drugs, the targeted drugs, the tyrosine kinase inhibitor that would change the survival. But equally important or more important was the fact that the patients that have long-term survival at distance, they had surgery for the lung cancer that developed in both lungs. So they have surgery in both lungs. So surgery was in fact the intervention that had the highest association with survival at distance. So that's why I've been recommending, since then I did something like 12 years ago, I did the study, and I've been recommending to my patients, if possible, to have oligometastatic disease removed. And I have now several patients that are following me, here in the clinic. They have long-term survival by using this type of idea that oligometastatic disease is a different type of phenotype for cancer. So you have to be as aggressive as possible, surgically wise.
Viruses that cause cancer, what can be learned from their mechanisms, and how do they do it? In HPV and hepatitis, et cetera. What do you know about that?
Yeah, so in HPV in general, the viruses are interfering with this TP53 molecule. So what is interesting, though, is that there are approximately something like maybe 60,000 new cases of head and neck cancer per year, and the HPV cases are increasing. What is interesting is that you have only something like maybe 0.01 of people that are infected with HPV that will develop cancer. So yes, we know now that you have these molecules, this P16 molecules that are really interacting with the with the P53 protein, and we know how the mechanism of action. But the real question is, how come you have only such a minority of patients infected with HPV that develop this cancer? And also another fascinating question is, in head and neck cancer, why does HPV affect only certain areas? HPV tumors in head and neck are really only in the base of the tongue and the tonsils. They are not present, on the anterior tongue. They're not present in the mouth. They're really not present in the larynx or pharynx. So it's only this location that is very, really high in immune cells where it develops. So it's totally counterintuitive. You're saying, okay, you have this virus, it's affecting the TP53, but why would it only there develop cancer and not affect some other areas, So, we know some things about this. We know something at the cellular level, at the genetic level, but there are many other things that are not known and questions that are definitely not answered.
Well, very good, Doru. Last question: What do you see as an advancement of cancer knowledge in the short term? Are there any promising avenues that you're looking at that you think in the very short term, 1 to 5 years, that will come to the clinic, or it's going to be a much longer haul than there?
Yeah, so 1 to 5 years, I really see the second wave of immunotherapy combinations that will become standard of care. We know now the checkpoint inhibitors, and we're going to have the data from combining checkpoint inhibitors with other ways to stimulate the immune system. So in the last 8 years, checkpoint inhibitors have been occupying more and more of the therapeutic arena in cancer treatments. And I think this is going to continue for at least the next 5 years. And then, of course, there will be more and more this type of ideas that I presented with the organism type of targeting and also influencing not only the cancer cells or tissue per se, but influencing also the other organs, like, for example, the liver. Can we make livers that are really not a good host for metastases? Also, anti-metastatic treatments, because at present, we really don't have any directed therapeutic tool against metastases. So we've been developing all these, chemotherapies, target agents for growth, for the hallmark of growth, but we don't have anything for metastases. So this is something which is badly needed. And, I hope that really in the near present, we'll also have that, anti-metastatic specific agents, which do not exist now.
Okay. Well, very good. Doru, thank you for coming. Where, for listeners, again, where can they find your particular research? Where can they contact you?
Yeah, sure. I do not have a website that is mine. There is no dorupaul.com or anything like that. But if they're going to just put on Google my name and then Cornell, On my Cornell page, there is, a list of the articles that I published, and also this idea of the system of cancer. If they're going to say Doru Paul and systemic hallmarks of cancer, they're going to find really a comprehensive paper describing a lot of the things I mentioned and some more.
Okay, very good. Doru, thank you for coming back. It's always good to talk to you, and you have tons of experience, so I appreciate it.
Thank you very much. Thanks a lot.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.